Hello, I'm Rebecca Jane Austin-DATTA, and this is my presentation on the multi-state outbreak of Salmonella mbandaka in 2018. This recording was prepared for the special topic presentation requirement for the PHC 6937 class, Environmental Ecology of Human Pathogens, Spring 2019, led by Professor Afsar Ali. During this presentation, we will discuss the definition of disease outbreak and the problems and opportunities that arise from them. We will explore the 2018 Salmonella mbandaka outbreak, which was linked to Kelloggs cereal, and discuss some questions that come to mind when you read the reports about the outbreak. To answer these questions, we will consider features of Salmonella in general and Salmonella mbandaka in particular. For example, the geographic distribution, ecological niches, survival adaptations, and the zoonotic pathogenesis of the bacteria. And finally, we will take a few minutes to reflect on future implications. The World Health Organization defines a disease outbreak as the occurrence of cases of disease in excess of what would normally be expected in a defined community, geographic area, or season. The CDC says that a foodborne disease outbreak is the occurrence of two or more cases of a similar illness resulting from ingestion of a common food. Local health authorities who suspect there may be a food outbreak are mandated to report these cases to the National Outbreak Reporting System. However, this is actually a voluntary procedure, so it is possible in some cases, in some understaffed regions, do not get reported. Also, as you'll see in a moment, if cases are non-specific, or they're not severe, they also may not be reported. Now, although relatively few illnesses occur in the setting of a recognized outbreak, data that's collected during outbreak investigations provide insight into pathogens and foods that cause illnesses. So public health officials and regulatory agencies and the food industry can use these data to create control strategies along the farm-to-table continuum that target specific pathogens and foods. So I mentioned that it could be possible that Salmonella's non-specific symptoms could cause underreporting of cases. So, for example, I really feel strongly that there is no guarantee that every case of Salmonella mbandaka was reported in 2018. So most people infected by Salmonella get diarrhea, fever, and abdominal cramps about 12 to 72 hours after they've been exposed to the bacteria. So symptoms may last up to 7 days, but the symptoms usually go away. No treatment is required. And most people would not be so sick that they would be willing to spend money on a copay or a doctor's visit. Therefore, they're not going to get put into the system and get reported. However, some people do get super sick with Salmonella, and the people who are more likely to have a severe illness are children under 5 years of age, adults over 65 years of age, and people with weakened immune systems. So in those cases, those people may need to be hospitalized, and Salmonella can cause death. However, I found very few cases where people died of Salmonella mbandaka. The Centers for Disease Control and Prevention, the CDC, say that there were 16 food-related Salmonella outbreaks in 2018 in North America. The Salmonella mbandaka outbreak was the 10th outbreak that year. It started in March, and by September, 135 cases had been confirmed by lab tests. Now, we discussed how Salmonella is likely to be underreported due to non-specific symptoms. the CDC estimates that for every reported case, 29.3 cases of Salmonella are unreported. And approximately 7% of Salmonella samples don't ever get their serotype decided upon. So this suggests that according to the 135 lab-confirmed cases we know about, there could have been over 4,000 cases of Salmonella mbandaka that year. You can see on the case count map that 36 states were affected, with California, New York, and Pennsylvania bearing most of the burden. Epidemiologists found that all cases reported that they had eaten Kellogg's Honey Smack cereal when they gave retrospective interviews to the epidemiologist staff. Now, it's a good thing that nobody died of Salmonella mbandaka during the outbreak. However, at least 34 cases were hospitalized. There we go. The age range was 1 to 95 years old, and the median range was 57 years. Now, interestingly, almost 70% of the cases were women. And I don't know if that is a reflection of the different eating habits of men and women, or female susceptibility, or females' willingness to report, or whether it's something to do with the bacteria itself. And we're going to come back to that later on in the presentation. Now, I said to you that Salmonella mbandaka is rarely associated with deaths. However, there is a case report of a serious mitral valve endocarditis in an immunocompromised 69-year-old male associated with the Kelloggs outbreak. When I was researching material for this presentation, I was very puzzled initially by the date of the Food and Drug Administration recall notice for Kellogg's Honey Smacks. You'll notice here it says June 14, 2018. Now, the epidemiological curve on the CDC website showed March 1 as the onset of first case of illness. So June 14, 2018 was 3.5 months after the first case, and that seemed like a dreadfully long time to be having a recall notice. However, it has been clarified to me that although the date of onset may have been March 1, for that first case, it could have been weeks later that it was lab confirmed and reported. So this flowchart from the National Outbreak Reporting System, NORS, that I showed you earlier, shows all the steps that must be completed before a recall can happen. And because it's the CDC, you can be very sure that the data has to be checked super thoroughly before it's made public. And before a recall can be issued, you have to do the legwork to find out for sure and get evidence to prove that a specific place is the source of infection. So investigators at this stage, where the green oval is, interviewed as many of the people who had been sickened by Salmonella mbandaka as agreed to be interviewed. And it was at this stage they found that all those sick people recalled eating Kellogg's Honey Smack cereals. So my forays into the FDA documentation related to the Kellogg's outbreak also raised some questions in my mind. It was clear that the manufacturers, Kerry Inc., were well aware that their processing plant had tested positive for environmental Salmonella mbandaka, yet they continued to produce and ship cereal for 18 months, even though all the time they were finding the environmental contamination. And this really puzzled me. I wondered why Kerry Inc. were having the problem of not being able to clean their factory, and whether they thought that because bacteria needs moist conditions to grow and survive, contamination of dry cereal, which they were making there, would not be a problem. However, once I started looking into Salmonella in general and Salmonella mbandaka in particular, I realized that these questions had tricky answers which may have long-term global implications. And I don't think it's too dramatic to say that even though this outbreak made 135 people very ill, I think that we may find that that was the calm before the storm. The CDC says that every year in North America, Salmonella causes 1.2 million illnesses, 23,000 hospitalizations, and 450 deaths. And the World Health Organization estimates that 59,000 deaths globally occur every year due to Salmonella. The CDC says that 1 million of the cases in America are food related, and World Health Organization estimates that 86% of cases worldwide are caused by food. So what's going on here? Why have I got a picture of chickens for you? The reason is that poultry are often reservoirs of Salmonella bacteria. So Salmonella bacteria are facultative, anaerobic gram-negative bacteria which have peritricious flagella. They're rod-shaped and between 2 and 5 micrometers long and 1/2 and 1.5 micrometers wide. Salmonella are divided into 2 species, Salmonella enterica and Salmonella bungori. Now, there are 6 subspecies of Salmonella enterica, and one of which is confusingly called Salmonella enterica subspecies enterica. The subspecies are divided into serotypes based on unique combinations of antigen molecules and the pattern of flagella on the bacteria surface. Serotypes can be further divided into strains based on their genetic profile. Now, to explain how complicated this is, there are over 2,500 identified serotypes of Salmonella, of which 1,500 are in that subspecies enterica, including Salmonella mbandaka. So using PCR, followed by multi-locus sequence typing, researchers in Poland found that Salmonella mbandaka has at least 6 genetically different strains, which are characterized by their biochemical profiles. In other words, they're going way down and looking at DNA of these bacteria, which is absolutely amazing. And what they have found is that some serotypes of Salmonella share genes, probably because they all come from a common ancestor. But for example, there's an 860 kilobyte inverted gene sequence found in 2 mbandaka strains that are also present in other Salmonella strains. Salmonella mbandaka is a non-typhoidal Salmonella, and it is considered an emerging zoonotic disease. It is named after a city in the Democratic Republic of Congo, where the serovar was first isolated and recognized in 1948. According to the CDC, human Salmonella mbandaka was not reported in North America until 1978. It is interesting that for 26 of the 34 years, 1978 through 2011, there were more female cases reported than male cases, which mirrors the data that we have in the 2018 Kelloggs cereal outbreak. Salmonella mbandaka has been found in places as far apart as Australia and New Zealand, China, India, and Europe. Salmonella's preferred ecological niche is the intestinal tract of warm-blooded animals, which leads to fecal contamination of the environment and of water. But Salmonella doesn't typically live in water. If you find Salmonella there, it's indication of fairly recent contamination by something pooping in the water. So Salmonella mbandaka has been isolated from agricultural, domestic, and wild animals and birds. It has been found in live fish, both in the ocean and in aquaculture, and in live shrimps. So this indicates that Salmonella mbandaka bacteria has adapted to live in a niche not favored so much by other bacteria. And although it's found globally, Salmonella mbandaka has different species distribution worldwide. For example, in the UK, the incidence in chickens and cattle is 65 and 20% of total cases respectively, and it's not found in UK pigs. While in the United States, chickens account for a quarter of the incidence, cattle about another quarter, and pigs 14%. One of the earliest animal outbreaks of Salmonella mbandaka was recorded in the United Kingdom in 1981, and the source was determined to be infected vegetable fat feed, which had been given to 200 cows. 28 of those cows had feces which tested positive for Salmonella mbandaka, but had not become clinically ill. And all the cows that got sick self-cured within 4 weeks with no treatment. The feed-associated mbandaka strain was super fast growing. A single colony was incubated in bactotryptose broth for 18 hours at 37 degrees Celsius, and it produced 10 to the 9 bacteria. So, whereas some Salmonella cerevars only affect one species, Salmonella mbandaka is a cross-species pathogen. It can be passed from animals to humans, so it's a zoonotic pathogen. There have been 2 recent outbreaks in the United States where flocks of backyard chickens passed Salmonella mbandaka to their human carers. Both direct and indirect contact with infected birds and animals can spread Salmonella mbandaka infection. Salmonella from feces can be carried on dust particles to areas where people may not expect to find Salmonella. For example, a significant number of human deaths from Salmonella mbandaka occurred in a rural population who were living nearby a commercial poultry factory in Poland in the 1990s. This outbreak spread Salmonella mbandaka to other parts of Europe, and the cerevar remains common in Poland today. For example, it was the cause of salmonellosis in 32% of human cases in a specific province in Poland. Salmonella mbandaka is also common in Korea, where it is found in 34% of chicken flocks that are tested for bacteria. However, Salmonella mbandaka is less common in poultry in Canada. Typically, just 10% of chicken houses have environmental evidence of mbandaka in Canada, and the cerevar is only a minor issue for Canadians, with Salmonella mbandaka causing less than 1% of Salmonella cases in 2011. So humans can become infected with Salmonella mbandaka by zoonotic methods, but also from eating undercooked meat and eggs which contain the bacteria, or by eating food that was cooked properly, but then it got environmentally contaminated after it was cooked. So outbreaks have been associated with eating items which I would not normally associate with bacteria because they're too dry. So we've discussed the Kelloggs cereal outbreak, and other outbreaks have stemmed from infected tahini paste, peanut butter, sesame seeds, and amazingly, dried salted fish. Because a bacterium is at least 85% water, I was really shocked to find that Salmonella mbandaka could survive the desiccation process involved in production of dried salted fish. So down here in the bottom corner, I have a table showing you how the prevalence of Salmonella mbandaka is increasing in United States livestock. So the United States Food Safety Inspection Service, FSIS, tested for Salmonella mbandaka in cows and bulls and in heifers and steers. And for those of you who need to run to Google, cows are female animals which have had a calf. Bulls are the main animals which are used to do what cows do naturally. Heifers are female animals which have not yet had a calf, and steers are males who have been neutered, so they're not doing what cows do normally. So you can see here that when FSIS tested cows and bulls and heifers and steers in the year 2000, they had a 2.5 and 5.26 prevalence respectively. However, both categories increased 10 years later in 2009. And you can see that for cows and bulls, so the mature animals, there was a 300% increase over 10 years. And in juvenile animals and the neutered animals, there's a 200% increase. So as I mentioned, this increasing prevalence in beef cattle in America is a problem because, as I said already, we can contract Salmonella mbandaka by eating food which is not cooked properly. So when you're looking at American livestock, it may not be an obvious problem that Salmonella mbandaka is present in a herd of cows, because Salmonella mbandaka does not always cause an illness in the animal. It has been isolated in healthy cattle. So thinking back to the 2018 Kelloggs cereal outbreak of Salmonella mbandaka that was traced back to environmental contamination at the factory, I have some understanding now where they may have had challenges eradicating the bacteria. So Anandino reports that a problem faced by facilities contaminated by Salmonella mbandaka is that the bacteria is very hard to eradicate. Acid conditions, which you might use when you're trying to clean, promotes upregulated bacterial growth. Biofilms of Salmonella mbandaka are resistant to chlorine, and dried out or desiccated bacteria can survive in environmental dust for over a year. It's not possible to detect dormant Salmonella mbandaka with current detection methods that you use in a routine process. So routine checks for hygiene, you could easily miss Salmonella mbandaka, and you might think you cleaned something just fine, but you didn't. Now, we've known for some time that Salmonella mbandaka, when it's dormant, when it can be carried on dust, and it can infect poultry via inhalation. So if you have got dust flying around the place, that is also a problem. So like most other serovars of Salmonella, Salmonella mbandaka is not particularly well studied. So outbreak investigations and follow-up studies have helped characterize features of the serotype, including information on how this particular serovar has adapted to survive many harsh conditions. For example, Salmonella mbandaka is resistant to dry conditions, so the desiccation is fine. So that's probably how Salmonella mbandaka became discovered on the continent of Africa. So even when the transport medium has been exposed to high levels of salt, which through osmosis will pull all the water out of the bacteria, researchers reported that after an outbreak in 1997, the CDC found viable Salmonella mbandaka bacteria in imported dried and salted fish. And it wasn't like on the surface of the fish, it was in the fish. And alfalfa sprout seeds were associated with another outbreak in North America. And those seeds remained contaminated with viable Salmonella mbandaka colonies, even after heat treatment up to 85 degrees Celsius, or after 11 months refrigerated storage at 5 degrees Celsius. But I do have good news that aqueous treatment for 10 minutes with 2,000 parts per million of free chlorine using calcium hypochlorite did kill the bacteria. So the end of the big long story here is Salmonella mbandaka is really hard to kill. Okay, so for those of you who know me a little well, you will not be surprised that I found a place to put molecules in my presentation. You know they're my favorite thing. So we have these molecules here because Salmonella mbandaka is starting to display multi-drug resistance. So Salmonella in general is now becoming considered a superbug. And Salmonella bacteria have multiple survival techniques, including development of antibiotic resistance and evolution of new serotypes. For example, Salmonella lubbock, a serotype discovered in 2015, is believed to be a combination of Salmonella mbandaka and Salmonella montevideo. Now, Salmonella mbandaka has been found to have integrons in its genes, intracellular mechanisms which appear to be responsible for helping the bacteria develop antibiotic resistance. So you may remember that Barbara McClintock won the Nobel Prize a long time back for finding transferons. They call them jumping genes that imparted special properties to heads of maize. So that's the plant kingdom. These integrons, they are associated with bacterial genes, but they're along the same lines of transferons. So it's quite concerning that despite its recent emergence and relatively low, if widespread global prevalence, Salmonella mbandaka in humans is resistant to quite a few antibiotics. Ceftriaxone, amoxicillin, clavulanic acid, trimethoprim, sulfamethoxazole, and it can cause persistent infections. Salmonella from poultry in Poland is resistant to cephalosporins and fluoroquinolones, while Salmonella mbandaka found in aquaculture fish grown in wastewater in Brazil and wild seafood from Indian oceans were resistant to tetracyclines. So did you know that that was a thing, that they are having aquaculture, so farmed fish, farmed seafood, and they're taking the effluent that's coming out of wastewater treatment plants and putting that in where the fishes are swimming. So any one of you who knows about how it's really hard to take pharmaceuticals out of wastewater effluent coming out of those wastewater treatment plants, just think the sea of pharmaceuticals that those aquaculture fishes are swimming in. So personally, I don't think that's a good idea. Anyways, where are we? Okay, so some researchers are looking at these 5 drugs which we have found that Salmonella mbandaka is resistant to, and looking at the other drugs that it's resistant to, and they're saying, okay, it's not great, but the good news is that Salmonella mbandaka is still considered treatable by antibiotics compared to other types of multi-drug resistant Salmonella. So in other words, Salmonella mbandaka is not the worst multi-drug resistant bacteria. However, I still think it's kind of a worry, and it just goes to show that it's really important that public health practitioners encourage people to use antibiotics responsibly, and we need to support funding to develop other types of antibiotics so that we're ready for when this one rolls. So in summary, this presentation, we've discussed the Salmonella mbandaka 2018 outbreak, which was associated with the Kelloggs cereal. We've discussed how disease outbreaks are obviously problematic because they cause people to be sick, but it also gives researchers the opportunity to dig into those particular cerebrals and find out more about them. We looked at Salmonella features in general. We looked at Salmonella mbandaka in particular, and we discussed the future implications of increasing antibiotic resistance. And I'd like to draw your attention to my reference link. I ended up with 36 references, and if I squished them down into teeny tiny font, I could fit them onto 4 slides, and I thought that was crazy. So in the discussion post, I am going to put the document with all the references. And also, if you're watching this presentation and you don't have access to those document links, you can see the bit.ly link there that will also take you to the document. So thank you very much. I hope you enjoyed listening to my presentation, and I hope we haven't gone too far over our 25 minutes, but never mind, it's fun. Thank you. Bye-bye.